DNA repair and its significance. Damage to DNA can be caused by various chemicals and physical agents. Introducing mismatches in DNA double helix may lead to a permanent change in the sequence DNA. This may also caused by errors in DNA replication that goes unnoticed by proofreading, that is called mutation. A mutation or collection of mutations can make a cell malignant, result in a cancer. Mutation also alter the phenotype, and often they are either lethal or at least produce unfavorable phenotypes. When the mutations occurs in cells producing gametes, they may be passed on to the next generations, resulting in variations among offspring. When a mismatch is there, double helix is disordered. For example, UV radiation can covalently link two iron thymine bases, distorting the DNA molecules. One of the two copies of DNA molecules generated by these distorted sequences carries a permanent change in the sequence, a mutation. Such distorted locations are usually recognized and repaired by the DNA repair mechanism before it becomes permanent. That reduces the risk of amplification of mutations. Because DNA repair is very important for the survival of organisms. A large number of DNA repairing enzymes are found in various organisms. Such enzymes may cut off the mismatched sequences in the damaged strand, replace with correct nucleotides. The cutting, excision, is done by nucleus. Filling the gap using the correct strand as the template is done by a type of DNA polymerase. This is called nucleotide excision repair. DNA ligase completes the DNA strand by sealing it with a phosphodiester bond.